Yeah, I've invited Rob to join. Chair's privilege, I've invited an extra panelist member, so given the talk, we can expand the questions. So we're going to we've got about 45 minutes to do this, so we'll try and finish on time. First things first, I'd like to introduce myself. So I'm Dave Brown, so I've been doing structure-based drug discovery for over 30 years in an industrial setting. I've done that for large pharma, I've done that in contract research organizations, and I've also done that in biotech. So I've got a sort of a good appreciation of the sector. And I think the problem, as Rob's just shown, the problems we have are multi-parameter problems that we are always trying to optimize using multiple methods to improve our chances of success. We do not know exactly how to make a good drug. I think that's the starting point. And the use of the word ligand here is a good use because we don't necessarily design good drugs all the time. We start off by designing good inhibitors or ligands for the target. And I think there have been some certain advances recently around AlphaFold, and this session is really to talk about how that's changed the way we operate and how industrial structural biologists and biophysicists are using that data or using those tools to improve our chances of being successful. So first of all, let me introduce the other panel members, or let them introduce themselves. Maria, do you want to introduce yourself first?
I'm Maria. I'm from Astex. So I'm a machine learning researcher in the computational chemistry group. I'm mostly working with cofolding and docking, and historically I was developing scoring functions for docking. So not that much of structural biology background.
But she's being polite there. Astex were probably one of the leading groups in sort of championing fragment-based drug discovery when it first started, which is, as Rob alluded to, soaking very small, soluble, high, relatively low affinity, but bang for your buck, highly efficient ligands into crystal structures. Chung?
I'm Chung, and I lead the structural biology and biophysics group at GSK. I've had most of my career at GSK, but I have done lots of things as well as structural biology. So I started off as an NMR spectroscopist, so I don't believe that there is a structure. I believe that there is an ensemble of structures that are in dynamic equilibrium. I've done a lot of biophysics and now crystallography and cryo.
I'm Frank van Delft. I'm at the University of Oxford and at Diamond Light Source, and I've been focusing on very high throughput structural biology, especially how to generate ligand co-crystal structures. So we set up the XChem facility at Diamond, which I think you heard about earlier, and are very interested to see how this can be used to push forward predictive methods, which includes this AI stuff, but more broadly, how you can actually make it useful. Do you need to introduce yourself again?
I don't think so. I think I know you know.
Hopefully everybody tuned in for Rob's talk, so he doesn't need to reintroduce himself. So I'd like to start off in terms of the panel discussion by saying the obvious question, why do we actually need liganded structures? And I'm going to ask Chung to lead on.
Okay, I thought that I would start by saying we have an assumption. As all of us structural biologists, I guess we think that because we're interested in human biology and therapeutic targets and modulating disease, that one framework of thinking about how best to do this is to start with the molecular. We know that interaction of our ligand, whether it be a drug or a small molecule, with a target or a protein is necessary for any modulation to occur. I think the assumption we have, which I think we saw should be questioned, is that just by understanding the protein ligand structure, we can understand mechanism and function. And in terms of experimental data, I would say that even if you could predict with infinite accuracy the experimental structure, one, there isn't an experimental structure. Two, I think it wouldn't be sufficient to be really helpful to us in the end. So one question I think that we should think about is, are we getting as much information from our experiment as we could if AlphaFold was infinitely accurate? And I think that is perhaps where the value experimental structures lie.
You want to pick up on
I think the other aspect of it is what Chung you were saying earlier about, as an NMR person, you've got a completely different perspective, which is you're talking about equilibria and lots of complexes. And I think that's what my eyes have been opened over the years of when, as you get more and more structures, it's not a structure, it is really a complex. I'm always struck by, back in my PhD in the late 90s, when I was part of a project where there were 3 or 4 competing groups looking at human growth factors, and they were all publishing at the same time, and one had this and the other one had that bound, and they were arguing about which is the correct one. And you look back and you think, well, that's complete category error. All these things are collectively informative, and I don't think anybody would ever have a bun fight in the literature about this anymore. They would simply say, here's a snapshot of it. And more to the point, do we routinely make many snapshots, and can we achieve many snapshots that are collectively informative to the model? I think that's really where it's going. So the other point that I've been seeing, watching medicinal chemists in these antiviral projects we've been part of, is that if you have one structure, you might start saying it's interesting. If you get 10 structures, you start saying, I'm not understanding this anymore. And then you get 100 structures, you think, no, I've really lost the plot. It's only when you get many hundreds of structures and they somehow feed into a model that you can maybe start making sense of it. So one of the challenges I see at the facility side is how can we help people routinely do these very large numbers of structures and somehow bring them together?
And I think, Rob, you said in your talk, that these tools are quite good at, maybe the ones we've tested like bolts, and now probably AlphaFold 3 available. So just for people aware, industrials, unless they have a particular agreement, will not use AlphaFold 3 because of patent issues around that. But in terms of those tools, the predictions of the poses are becoming quite accurate in terms of modeling terms, representative of many of the X-ray structures that they've been trained on. But it's the ranking of the binding energy. And at the end of the day, so I think there was a question in your talk about, where did you find your starting points? And you mentioned virtual screening. So for this audience, can you just talk a little bit more about sort of the computational side and the needs in that?
In virtual screening, so what we routinely do as well is actually, if you have a protein structure, what you can do is actually dock a vast library of compounds in there and see if that gives you any ideas on what molecules to synthesize, and ultimately that is what the chemistry wants. I think an experimental structure, I'm an experimentalist, so I'd always prefer that. But if you haven't got that, it's as long as chemists have starting points. But the thing is, you always use, whether it's a docking, an homology model or whatever, or an experimental structure, or a collection of structures, you always have to correlate it with other data, and it's just one view. A structure is just one view, or a set of structures are a couple of views, but you have to correlate it with other data and check that it's binding to your target. That's where biophysics comes in. But ultimately, what you're interested in is how your drug behaves in a cell and in a body.
Maria, would you like to add more in terms of the screening or virtual screening and ranking of compounds, in terms of where we're up to in terms of state of the art with these types of tools, and not just the AlphaFold's tools, but the actual other computational experiments, computational tools that are used to rank and score these docs?
Yeah, I think, well, in terms of the AlphaFold tools, and in particular, for example, the bolts tool that had the affinity prediction head, were quite not there. We did good internal benchmarking of folding, mostly for post prediction, not for the affinities. And yeah, we're not yet there. But in general, the idea of getting the affinity from a single snapshot that is predicted based on X-ray only experiments, this is a very strong thing to wish to happen. Yeah, so I would say that in some cases, even 2D based virtual scoring functions for virtual screening, models for virtual screening. Sometimes they work even better than 3D, which means that we're not catching something important in our models. So physics-based methods is probably what we are currently using to get the best assumption about affinity, FAP.
Maybe a good example some people might know about is if you get a liganded structure into proteins that are very similar at the active site, if you look at the active site, it can look almost identical, but the affinities can be somewhat different. So what that suggests is that the way that we are trying to interpret affinity by just looking at the structure, we're missing something.
I have a question for both Maria and Rob. What you know from the AI field, because some papers last year, about that these models are predictive in islands, more or less. You can sort of have the local predictive volume of generalization, and then that's a feature of both the binding site and the ligand. So it's something about the interactions, and it's not enough to simply say you've got the pocket characterized. You have to. It's the kind of functionalities. So if you get a bunch of hits or you try and compute them, how do you go about working out which of these interactions that you observe are maybe the ones to take seriously? I was struck by your examples. You don't have super potent things. They're like 100 micromolar, but still you have a decision point there that you go after that thing anyway and then keep evolving. I mean, how do you attack that problem? Maybe Maria first.
Maybe Maria first, yeah.
Yeah, to be honest, I'm not sure that I can answer a question because I'm not a trained computational chemist. I don't know how much. I don't know how much of the computational tools and how much of the personal experience is used there. So.
Yeah, I think I have to say, of course, we use computation and see how, but essentially what we did is these were projects that were more experimental. We looked at analogs and we tested them in biophysical and biochemical assays. And the ones that were more potent than the others, we soaked again. Then you, of course, get into a structure-based drug design, and you try to use the computer to actually fill out parts of the binding site, etc. But it was fairly traditional, I would have thought.
Yeah, I would say, I think you've got to realize that people are always pragmatic in terms of what they can do. So a ligand-bound structure gives you a hypothesis of what might be key interactions. The computer modeling sort of reinforces that, but then you actually have to synthesize follow-up compounds to test that hypothesis. So, the computer accuracy is not enough to say that this is definitely the right thing to do. And actually, when you work within you saw on Rob's slide just how big these teams are. You might have a team of, if you're lucky, 20 chemists synthesizing compounds for you. They're going to make the stuff they can make. If there's a simple analog they can make, they don't need to dock it and design it cleverly. They're just going to make it. What we should be using structure-based drug design for is that more bigger step hypothesis change in terms of if it means they've got to change their synthetic route, but there's enough value from these computational models to say this is worth your investment of an extra 3 weeks of your time to change what you consider to be your core of your series. That's where structure-based drug design can have a massive change on influence on it. A lot of what we do is actually massaging and giving them confidence that what they're doing is going along the right line. Where we really want to apply really turn the handle or change the mode is this structure really says there's a different hypothesis that you guys aren't currently exploring and you need to explore it. And I think that's, for many years of experience, the biggest challenge we have. And the question around when do fragments come in, if they're too late, people are already down the road. It's like the oil tanker. It's difficult to turn them once you get going. And actually fragment splicing is a good way of actually later stage programs, they get stuck. You do a fragment screen and it actually shows a way to change something that they wouldn't have thought about changing because they thought it was integral to their binding.
Yeah.
Yes. So I guess one of the things that Frank said was, can you look at interaction saying, oh, this is the interaction that's important? I think one of the things that makes it really tricky to predict binding is that binding is delta G. That is the Gibbs free energy for everything before it's bound to everything after it's bound. And just looking at the active site, I think we're missing a lot of things. So this thing that I said about selectivity, the reason why just looking at the active site doesn't work is that you can get waves of, slightly more flexibility, slightly less flexibility for a protein, because the protein is designed to dissipate energy. So that's what it will do to different extents in different proteins. So it may be the third level or the fourth ring out that you need to look at, which means that prediction will always find it difficult. And that's why experiments sometimes helps us, because we don't have to guess it. We measure what is actually happening rather than trying to weigh up all of these energies.
I think that's a really, really good point. And something else that Rob alluded to, that quite often we're working in a very reductionist system. We've made a construct which is part of a protein, not even the full protein. It's not in the right environment. So why do we believe it's going to be that predictive of designing a drug that's going to work in a cellular environment? And actually, that's where a lot of things in terms of combination, people say cryo-EM, is that killed off X-ray within industry? No, because we need the high resolution from the X-rays that might give the definite detail within a construct with all the local water networks, which are key in terms of drug design, local perturbations, but then you need to look at in the bigger sense of the bigger molecule or the macromolecular complex. If you can go to cryo-EM to do a larger scale system, that this translates, and what you're seeing in your X-ray actually is more likelihood translating to the cellular activity. Thanks.
So one thing that struck me, well, as we've been setting up this open bind consortium, trying to work out what data to measure is one of the big opportunities that seems to come from the machine learning approach, is this cross-fertilization. So if you've measured one set of kind of interactions or pockets, it may be the model is the only way to know whether that could be informative for a completely different type of pocket and different set of chemistries, because it's not easy to work out by inspection or physics methods. That doesn't mean a lot of data needs to be measured. And I should emphasize again, there really isn't very much data at all out there right now for learning. So the estimates that I keep hearing is 20 to 30,000 pairs of structures and some kind of activity. And the high quality things are maybe as low as 2,000. It's really very, very small amounts of data. So it's lots to be measured still. But I think the other thing that struck me now for a long time is that how little of what's really measurable or measured gets translated into the model. If you just think of what we do, we put these little atoms down and we put an isotropic B factor onto it. And then we say, okay, off you go. Now use this in computations. And there's so much more signal, the R factor gap, which structural biologists know and ignore. And we haven't cracked it. And what you do with many models measured over hundreds of data sets, we just don't have a way to articulate that. And I think there's a big opportunity to sort of bridge that gap of how it can be learned and how we should, as experimenters, feed that through to those models.
Yeah, and I think coming back to that binding affinity, and you mentioned FEP in physics-based models. Well, if you're measuring an IC50 in a cell, that's not a direct binding energy. So actually the biophys, and it's interesting in terms of the way that the groups have evolved, particularly in industry, that the structural biology, the protein production, the structural biology and the biophys groups tend to be a group who actually understand that when they're translating data from an SPR or a thermal shift assay or some of that biophysical assay or mass spec affinity assay or NMR, that they're actually the same people working the same team that doing the X-ray structures and going, actually, there's a disconnect between the construct of the protein we're using or the conditions in which you're measuring your affinities. And actually, in other organizations, it was like, well, what do you link it in your own internal structure database? Do you link it to an affinity, a direct binding affinity, to help doing things like your large language model development as you're going forward? Which affinity do you use? Do you use the SPR affinity? Do you do a mass spec, a ligand displacement affinity?
And do you do the affinity in the crystallization buffer?
So I think there's an awful lot. And like coming back to Frank's point, we've been, as a community of structural biologists, we've been very good in creating the PDB, which with sort of the large amount of genomic data around in terms of sequences, plus the structural information has led to AlphaFold. We're now actually, with the computational methods and things like co-folding approaches, we can actually predict shapes that are adopted by both proteins and in the presence of certain ligands, what those shapes are. But actually, when it comes down to the affinity component, we're still a long, long way off for many of and it is a really big problem to try and solve. And it will only be solved by having the appropriate metadata stored in the right format and enough of that data to be able to take us to the next level. And I think that's sadly several years off, but I'd like Maria to comment on that in terms of data quality and amount.
Yeah, well, I think when people just started learning on affinities to predict them, they were kind of misusing the data, like mixing the assays with each other. But I think now people try to exploit a bit more like practices from statistics to do some sort of meta learning so that the model itself can try to learn that some assays are different and try to get some kind of information that is probably related exactly to the binding and not to some random correlations in data. Recently, I read actually an interesting blog post in which someone did some analysis on, I think it was Campbell data, and they found out that sometimes the affinity is even correlated with the name of the chemist who was designing the molecule. Yeah, because it correlates with the project on which they were working, but it also correlates with the modifications that the chemist would probably often do. So yeah, there are many biases that we need to consider.
Yeah, and I think just on that point, I think it's a really interesting point because you say you're not a computational chemist. If you're not a structural biologist, people like coming from a different field to our field it takes a significant amount of time to actually get the language right and to understand what each other are doing to actually get the best approach to solving the problem.
Yeah, I think when I just started, I really believed that we can reach something like 0.9 Pearson correlation with affinities. And then I found out that on like 2 assays from the same team, it can be less than 0.7. So yeah, the problem should be formulated also very, very carefully because the data is quite dirty.
Yeah. And Frank, I guess this is a challenge you're trying to take up within the open binding. Right?
I was going to say that it's the moment you start delving into it, it's really obvious that all this AI buzz, thinking AI is now displaced experiments is just rubbish, and we are a long way from that, and there's a huge opportunity need for improving the methodologies. But one thing the AI buzz does do is to make us maybe focus on where the real things are that we can develop as methods communities and sort of think harder about what we're not measuring or not capturing. And I think so it's productive in that sense. It's sort of flushing out these things we've been brushing under the carpet. And then when you start trying to systematize it by a model, like a learned model, you realize the model has gaps, and that's because we're not putting the right data out there. So I think maybe this really should be a strong message from the experimental community, people in this room, to funders and the buzzwords and people that are trying to mobilize money. No, experiments will matter much more than computation. Don't build new data centers. Don't build, fund huge cohorts of geeks that just crunch data. Fund people that understand the data and will systematically put it together and work with the people that know how to use them in learning. And it has to combine. And we've been doing this all these years, trying to somehow feed models and physics-based methods. But the integration of this is much more important now than it used to be, because now you can ask the model directly, which data should I measure now to improve the model? And we can iterate this quickly. And it's a huge opportunity for the field.
Yeah, I think one of the things that perhaps isn't well integrated is like the use of AI whilst we're doing experiments. So I've got an example, it's outside of structural biology, but we were interested in using some machine learning methods for HTX. And we wanted to draw some correlations. And one of the things that we found was that the signal seemed to, the largest principal component was which batch whether the what they showed us, and we tried really, really hard to standardize. And the reason that it was correlated with each batch was that in HTX, normally you take away the baseline because it's a difference experiment, and the baseline was more variable. So we had introduced, because our eye and the human eye is better at looking at difference, by making it visible to our eye, we'd actually introduce more error into our experiments. So we just dumped the taking away the difference, which meant that the way that we analysed the data was informed by how we were using machine learning. And that's something I see less of. Do people use the analysis of the data to say, actually, perhaps we should be doing this differently, and actually we should be doing the experiments differently, so it maximizes the information?
I think this also picks up on the meta point, which is you can generate data in the database and people can start learning from it. But if there's nobody that understands the data and in real time critiques what the model is doing, you will just generate models that predict rubbish. And I think so. This is why I think the experimental aspect of all these learning tools, and it's not just for structural biology, it's all the AI buzz. You know, I think it's in the next 5 years we'll establish this huge category error. Oh, there's enough data, let's just learn harder. It's just the wrong place to invest. We need people, experts like you just described, to say, "Ha, we've just completely misthought this thing."
Yeah, and I think coming back to there's, you know, I've been sat on grant committees in the past, and the modeling system, not with no experimental data link, never gets funded because you need to have that feedback mechanism. And you know, another example is generative AI in chemistry design, where the chemist looks at what the computer tells it to make and says, well, that one is impossible to make, or two, it's going to be inactive, so I'm not going to make it. But you actually have to have those people make the molecules that are synthetically tractable to actually retrain the model. So there has to be a feedback mechanism in these things. If you don't, a model will always be bad unless you give it the right data to train it. So, but it is a two-way street, definitely. So what I'd like to do is just open up the panel to the audience and see if the audience have got any questions, or are there any online questions for the panel? Do you need a mic? Gerard's got the question here. Thank you.
Yeah, Gerard Bricon from Global Phasing. It's a question mostly to Frank, because you've pointed out that there's not much data out there to learn from on the scale on which it needs to be learned. And then pointing out the importance of the quality of not just metadata, but also of the data themselves. So you have this very impressive project for the K04 beamline that should deliver 10,000 data sets a day for crystallography-based screening. What do you base your estimates of data throughput versus data quality, including the optimization of the quality of the samples upstream of the experiment itself, to be confident that you can deliver that sort of throughput with a useful data quality coming out at the end?
That's an excellent question. I think what one can do technically, and that's what we synchrotrons have been doing all along, is to just make sure the throughput of the measurement is possible. That doesn't in any way guarantee the quality of the measurement or the quality of the experiment, and that's never been true. I mean, we've been cranking up, throughput all these decades, and it's always relied on the user community, the scientists, to design experiments that are sensible. What one can do technically is increase the capacity. So I think there is a responsibility in two directions. One is that one not just develops the throughput of the measurements, but the if you like, the funnel that takes them in, and as if you like, curators or stewards of these facilities, we have to try and help the community to make things as sensible as possible. At the same time, you have to try and build a community of experiment designers that are much more holistic than simply saying, chug a bunch of crystals and throw in any old rubbish and see what comes out the other end. So I think that has to grow alongside all facilities. And I think it's been great in structural biology that it's kind of, I think we just look at the evidence, the PDB is an evidence that it has happened and can happen. But there's definitely a expansion of community need, and people that will use these very high throughput facilities will have to engage with how to make sure the experiments are correct. And I think the challenge as the throughput goes up is that your number of dimensions of data quality get more. It's not just enough to have good diffraction. You have to have good experiment design, good chemistry, diverse chemistry. All these tools have to develop in parallel. I think that's kind of where the community has a challenge. And it doesn't rest only with a synchrotron. It's maybe just a trigger for that, but it cannot solve the problem on its own.
Frank,
Yes.
Can I ask you a question on that then?
Sure.
So OpenBind is going to generate a lot of data. There is a possibility it can't generate enough data, even with all the resources. When will you know?
Yeah, I mean, I think this is the key one. We think of, I've still got this mental picture of islands of information or predictability, predictiveness. So for any such effort that do, lots of data, and that's not true just for structural biology, you need some sort of orthogonal calibration of usefulness. And so you have to have people that will use the tools actively and in a visible way to say, is the tool useful or not? It's not enough to put a blog post out or a tweet to say, look, we've got the new predictive model and now you can go solve things. You have to have projects that are visible and interrogatable for its applicability in real discovery. And so this is why one of the key things we're putting into OpenBind is you want partners that are active discovery people, whether it's closed or not. And you also want publicly funded discovery projects that make use of it. So people can go and look and say, oh, is it useful? And then that question of applicability is the key one, right? Have we looked at enough target classes, enough pockets, enough modalities? It's infinite, as you know. So it doesn't even feel like it's a closed question. It cannot be closed. It's probably going to be open-ended for the next 2 decades. Have we covered certain islands? Let's measure more data. So I don't think the facilities will go away as a need.
As far as I can understand,
Sorry, can you state who you are for the online people? Who are you? Where are you from?
I'm Shafat Islam from Helsinki University. So as far as I can understand, binding affinity, determining binding affinity is a dynamic process. And when we calculate it, we calculate from a dynamics of the whole protein ligand complex. But when we are designing our AlphaFold 3, Bols 3, or any other binding affinity based models, I don't see that the machine learning designers, they are not accounting for this. So I don't know, are they have they considered these dynamics things for their feature selection, or I don't know. What's your comment on that? Is my question clear?
Sorry. So you're asking about how do the holding model accounting for dynamics?
Say that again.
How do the all-atom models compensate for dynamics? Or
in the AlphaFold model?
Yeah.
Are you thinking about taking into account these dynamics in the AlphaFold model or in the Bols model or in any deep learning based model?
So, well, by design in AlphaFold model, it does not account much for the dynamics. I don't know how much are you familiar with the recent state of the art of how do people try to account for it? Because one of the ways is that when the protein is folded, the model gets lots of input from the multiple sequence alignment to know what residues evolutionary would be close to each other. And one of the ways to kind of alter the conformational ensemble that you get in the end was playing with multiple sequence alignments. subsampling them so that sampling results in different conformations. But again, this is not like really modeling or incorporating the dynamics. This is just trying to get a better conformational ensemble. With diffusion models, it samples a little bit more naturally, and some people, for example, tried to incorporate an energy proxy into the diffusion model, which then can be used to try to sample with that kind of energy proxy. But again, it is not clear if this gives a full dynamics, but maybe this will work. But it looks like this is the direction towards the field is going, to try to get more realistic conformational ensembles and then hope that having a good equilibrium ensemble converges to proper dynamics representation.
I'll just add one little thing. The data that might inform the dynamics just aren't there. There's you can model them, or you can try and do dynamics, but in principle, we do 1,000 data sets for a screen that could have enormous information about conformational variability. There's no way to capture that, never mind extract it. So what they're going to learn against? So I think there's another whole dimension of lacking data that can also be filled in.
Hi, Matthew Rodriguez, Diamond Light Source. So in crystallography, we don't just deposit a model, but also structure factor, amplitudes, unmerged intensities, and ideally raw diffraction data. And I was wondering, when you're collecting these pairs of structures and affinities, is there value in not just the IC50, but also the raw assay data and information about how you get from that raw data to the final results in AI and machine learning? Or do you just need the final data?
I mean, my assumption is that there will be value, and we need to capture it, and there's no database that captures it right now. So it's actually one of the big gaps we're trying to worry about, and I hope others as well. Like, just how do you encode it? At what level of raw data is it worth it? So of putting raw data sets out there, diffraction data sets may not be very useful, but for instance, structure factors, that might be, or electron densities at least, so there's these levels of abstraction, and it's not clear at what point you can learn, and the same is true for the biophysical measurements. I should emphasize not IC50s, that's an activity, but affinity, which is KDs, and there's different ways. You can K off, K on, or you can do your derived from thermodynamics. All of these have got sensograms or various readouts. So how much of that can you deposit? And the machine learning people have not even engaged with that, because they haven't had the data to even think about it. So there's a whole methodological ocean there as well. But I'm sure there must be something.
Can I comment that the ratio of K on and K off is only equal to KD in a subset of circumstances? I know people do that, but it isn't always true.
You must comment.
So I think that it's only true if it's one step and you're measuring the key step that leads to the KD. So I think some of these things, therefore, we need to really think about.
Yes, this is very a good point. One of the big conversations we've had is only been a few months is, what exactly do we want the models to predict? A KD is just a number, right? Should we have some sort of shape that's derived from dynamics or something? And what does the medicinal chemistry actually want to know when they are planning their chemistries? So I think these are just not well-defined questions. Even though they may look like it, oh yeah, I'll predict the KD because that's what I get in my database. What can you do with that?
Yeah, like going back to the experimental, to the structural experimental data, I think maybe in different aspects of the problem, it can help in different magnitude, because for protein prediction, especially for protein monomer prediction, if it is not disordered, I don't think much can be obtained from the data, but probably like for disordered bits or for predicting ligands when we don't have almost any additional information,
would help.
Yeah, I think there's an obvious thing that Frank mentioned earlier in terms of we expect these computational tools to be 100% accurate. Yet as structural biologists, we have probably a 20% error in our experimental fit to our experimental data, which we happily ignore on a regular basis. And coming back to what does the chemist want? Sadly, the current state of the art is they want one number, one structure, because they cannot work with, they want a unified number, not two different affinities. They can't deal with that. And that's where we as a group and that's where structural biology is really interesting. And coming back to where we started this discussion, we look at things from an atomistic point of view. We want to understand how molecules interact at the atomic level to better understand the interactions of how they may work in a cellular environment. And a chemist tends to, and we think in 3 dimensions and we think in biological sense. We span between the chemists in the drug discovery program and the biologists. And we've got a foot in both camps. And that really puts us at a strong advantage in terms of helping interpret the data we have and sharing it in terms of the drug discovery environment.
So Dave, maybe I can ask you a question. I guess all areas are using AI and machine learning. That includes chemists. Obviously, medicinal chemistry is a multi-parameter optimization, has always been. You were interested on the KD on the target, the KD on off targets, the biodistribution, all of those kind of things. And there is much more use of machine learning AI as for chemists to understand their breadth of data and not one number. Do you see that we could actually introduce more data to them? Because if we start saying that it's not one structure, there's actually 5 different structures and this kind of stuff, do you think that there is more appetite for more data, and what kind of data?
So I was trying to hint at there's an education process that throughout my career I've done with each generation of chemists to educate them in terms of the level of accuracy and predictability of what we're giving them, both in terms of the structural information, the selectivity of that can infer by different comparisons with other models or computational models that are derived in terms of deriving the next best molecule to make. It's coming back to what I said before, we can guide things. We will never be 100% right, I think. And I don't think we'll ever be 100% predictive, unfortunately, Maria.
I do think, though, there's an opportunity that comes from these formalisms that pool all the data. So because there's some systems which are quite well behaved, right? The binding site is quite a uniform conformation. You can bind anything into it, and you sort of see common behavior across things. And the other ones which are much more diverse, you've got these multimodal binding effects, and you don't have a single KD. But what you may find is that these well-defined systems can sort of cross-inform what happens in subsites or subsets of the process, and in a way help formalize it, which is quite hard to do by physics-based process, but at least can say, well, what's happening in this part of the pocket looks like what happened in those 5 other pockets. So maybe that's the model to use for that sub-pocket. And that's certainly the thing I've seen the chemists always trying to get their head around. How do we make sense of all the bits of the molecule and the binding site? And it's hard to tease apart. And maybe here's an opportunity that might come from these, if you like, all formalisms, like these machine learning approaches. And then maybe that will sort of auto or educate our chemists.
I think design is not done by an individual. There are individuals that want to design on their own, but actually it's become, coming back to your thing, a much more federated approach to design of drugs now, involving multiple people from multiple different disciplines to actually inform what's the right thing to do. And what I would say, this all sounds gloom and doom. What I've found in my career is that actually what we're able to do is the targets we're working on are probably more difficult than some of the targets that have been done in the past. We've taken, the drug discovery has been incredibly successful for global health. We've got more difficult targets now. We've got different mechanisms we're trying to use in different approaches to drug these less druggable targets, but we're still making advances. We're still putting new molecules into the clinic to help patients. And a lot of that, when I started, structure-based drug design wasn't part of the drug discovery process. It is now, it's an integral part, and will remain to be so. The more informed we can be, the quicker, hopefully, we'll get drugs to patients. But actually, one of the paradigms that we're doing is actually what we've also seen is the barriers to getting something there. All the things that we did at a late stage and had late stage failures, they've all been brought forward in the process by use of more advanced tools. And we have the success rates are plateauing, but that's a good thing, that we're actually able to maintain a success rate when the targets are becoming and the constraints of what we're working in are becoming more difficult. So I think just generally just wanted to wrap up and say, I don't think we'll ever get, everything will ever be 100% automated by a computer. But I think in terms of if we use these tools in the right way, we will maintain a competitive and useful role to play in society into producing new drugs. That's probably a good place to stop. And I'd like to finish by thanking the panel for their contributions today. And if you could put your hands together, then great for dinner.
Just before everyone leaves, if you've got a poster judges will be going around this evening. So please grab some food and head to your posters and talk about some science.
Thank you.